welcome. If you want to post in the chat maybe where you're from, that would be great. And then my name is Chris Herman, and I'm from 3D Molecular Designs, and I am going to moderate along with Rachel Mosey from 3D Molecular Designs. Welcome to DNA with a Twist, activating student questioning with an interesting twist on DNA modeling. Today our presenters are Karen Avery and Dan Williams, and I'm going to let them introduce themselves.
Hi everyone, I'm Karen Avery. I have a BA and an MS in biology, and I'm currently working on my doctorate at the University of Pittsburgh. I teach at the Pennsylvania College of Technology in Williamsport, but I spent the bulk of my career as a high school biology instructor.
Good morning. My name is Dan Williams. I'm a high school teacher from Shelter Island, New York. It's a small rural school district about 100 miles east of New York City. Before we get into our goals, I wanted to just highlight that you see that Karen is from university, or she's at the college level, and I'm a high school teacher. And what we're going to be modeling today is something that's so flexible, it can be used at both the college level and the high school level. In fact, in my district, I teach everything from 9th grade all the way up through seniors. So my freshmen come in working on these models. So for today's goals, we're going to be looking at fostering student questioning using the dynamic DNA and flow of genetic information kits. We're also going to be teaching DNA structure and function with the dynamic DNA kit. And of course, this is such a great model. We're going to show what else can you do using these kits that are out there. And after being so focused on the dynamic DNA, we're going to zoom out so you can see the whole big picture with the flow of genetic information. And we hope to get some hands-on activities for you all to play with.
So to get the conversation started, we are going to ask you to join our poll. We are going to ask you to tell us what makes a good model and how you use models in your classroom. So just what makes a good model. You can go to, and I think maybe you have to, that should automatically go to the screen for joining the poll. You can go to poll everywhere at, here we go, poll everywhere at Karen /Karen Avery 491. Rachel's posted the link in the chat. Or you can text Karen Avery 491 to 37607. And what we should see there are your answers. You folks should see my screen. We've got some answers coming in. So far, a good model looks like is something that's hands-on or self-correcting. Intuitive comes up twice. That's a good one. Engaging. Accessible to everyone is a big one. Engaging for students. Relevant. Absolutely. Representative, engaging, and hands-on. And so it looks like you folks are where Dan and I are when it comes to modeling. It needs to be engaging to students. It also needs to get their hands busy and somewhat intuitive with a little bit of our help. I'm going to stop my screen share and turn it back over to Chris, who's got this slide deck. And we're going to move ahead then to a couple of models that you may typically see. Oh, we got another one, hands-on and critical thinking, which is something that Dan's going to be emphasizing as he talks about this. So you may have heard the quote, all models are wrong, but some are useful. And what you see on this slide are 4 different models that we typically see as icons or in our textbooks or on worksheets. And this time using the chat, Dan and I would love for you to pick one of these models and tell us either how you would use it or what you like about one of those models. So in your chat response, just pick the number, tell us what number you're talking about and what you like about it or what you might use it for.
Take the purple.
Those answers go in the chat. Okay, so one gives a simple view of DNA structure for intro lessons to 7th graders. Tracy tells us that you would use different models for different concepts. Courtney says one's a good intro for coordinating colors and asking students what they notice. I like that one. Notice things are big. 2 is more specific. 2 for nucleotide structure. 3 for specificity of base pairing. And 4 for showing structure. 2 to show why base pairs pair the way they do. So as your conversation over in the chat box is elucidating for us, there are different goals for each of these different models. So you might use one model for one thing and another model for another. Dan and I are going to take a minute to have a little conversation here with each other to talk about how we use models in our classroom.
So for instance, Karen, what is a model and what form do models take?
I think of a model as something that takes either a really huge concept and shrinks it down, or something that takes a microscopic concept and blows it up so that we can manipulate it either with our minds or with a pencil and paper, or actually physically moving parts. So a model can be a lot of different things.
Is there such a thing as a perfect model?
Aha, no, there's not a perfect model, but there are unique models for unique applications. So what's your favorite model, Dan?
Well, I always like to think back to Matchbox cars. They were always fun when I was a kid. It looks like a car. It actually kind of acts like a car. And so it's fun and colorful and intuitive, where you can start playing with it, make little ramps and see how far it can go. And I'm sure in physics class, you can test all sorts of different things within it to see about velocity and movement and stuff. And yet it's not a full-size vehicle that's going to go smashing through your parents' window when you're playing with it in the driveway or something. So it's a very useful model and it's fun.
Oh, I like something that you added in there. There's low risk with using models. So you can try things out, see if they work, and you don't have to risk changing anything. Oh, we got a shout out for Kimberly from Kimberly, thanks. It's true. All right, so we're gonna be bringing you 2 models today. And the first model is the dynamic DNA model, which is going to show us one scale of DNA. And the second is the flow of genetic information model. Some of you have received a student kit for at-home use for the flow of genetic information. But the first thing we're going to start with is the dynamic DNA model. And we're going to show you a quick video to kind of get you oriented with that dynamic DNA model.
Hi, this is Karen, and I want to take you through a quick walkthrough of the dynamic DNA model. You can get this model as 2 base pairs. You can get it as 6 base pairs, and you can also get it as 12 base pair set. You can also, if you have a large class, you can get 6 sets of 12 base pairs, which would enable you to model replication and such, which we'll talk about a little bit later. I'm going to use the 2 base pair set to show you just a couple of small things. You can see that we can already go from the ladder model to the helix model and show those rotations around bonds. But in addition to that, we can take a second to get 1 base pair here. You can take these base pairs apart easily because there are magnets on phosphate and the oxygen on the 5' and the 3' end. But if we look at 1 base pair, you can see that CPK coloring is used. And that's really nice because this translates to probably a lot of your textbook documents or the documents that, or the images that you're using in instruction. Also, here we have a pyrimidine base pair, so you can see every single atom. You can see that the hydrogen bonds are magnetic, so you can model those weak attractions and unzip that molecule down the middle. You can also construct or deconstruct a nucleotide. This is nice because we know that nucleotides are not flat as they are on the diagram. And here, again, you can see every atom. We can pull off phosphate group. And you can also take off the deoxyribose sugar. Here's our deoxyribose sugar, and here is our nitrogenous base. So you can either construct or deconstruct those items. The bonds are just a little bit more robust between those. Those are covalent bonds, and it's important that we are able to show that they're a little bit stronger than our hydrogen bonds.
So before we get too far into this presentation, I know that usually when we look at the DNA structure, that already takes our brain into DNA replication, transcription, translation, all the central dogma topics, which are critically important. But I do want to take a minute to walk you through some additional ways that I use the dynamic DNA model with my students. First, to articulate the difference between a DNA nucleotide and an RNA nucleotide, and second, cellular energetics.
Now that Dan and I have gone through how we use dynamic DNA model to teach DNA structure and nucleotide structure, and how we are so impressed with how it elicits student questioning and how that student questioning leads to deeper learning, I'd like to dig in a little bit. I'm going to touch on DNA replication and transcription, but also why we could take a minute to talk about some really important differences in DNA and RNA structure, and also how I use dynamic DNA to teach ATP and phosphorylation and relate it back into cellular energetics. I'm going to advance to the next slide here. And in doing so, show you that if you have a classroom set of dynamic DNA for DNA replication, it's fairly obvious to see how we can use that DNA, dynamic DNA kit for DNA replication. We've got a model here that we can use to go from the double helix model of DNA and use our hands to untwist it. Students can then use this dynamic DNA model, pull apart those hydrogen bonds, and begin to fill in the nucleotides on the template strands. That's pretty easy. But when we go to transcription, we have a problem because these are deoxyribonucleic acid nucleotides. These are DNA nucleotides. And so how do we convert these DNA nucleotides to RNA nucleotides? Well, I call that going from flat to fabulous. Here, I always start with the model that probably a lot of us start with. This is the flat model that a lot of us use to teach nucleotide structure. We've got our phosphate group, we've got our deoxyribose sugar, and we've got our nitrogenous base. I hope I'm going to convince you of the ease and importance of differentiating this DNA and RNA nucleotide structure. I think a lot of times we have our DNA column and an RNA column, and we talk about DNA, it's double-stranded, RNA, it's single-stranded, which is a conversation for later, but not today. We have our deoxyribose sugar in our DNA nucleotides and our RNA sugar, or our ribose sugar in our RNA nucleotides. So why is that important? It only takes a second to go into that difference between deoxyribose and ribose and kind of solve that maybe slight discomfort students might have when they say, well, how is deoxyribose and ribose, how are they different? Let's focus on the sugar here. So I'm going to blow that up for you, and I'm going to tell you not to be afraid, that your students can number these carbons, and it'll set them up for success in their chem course later, as well as helping them understand why the difference between deoxyribose, and ribose is important. I think this sugar looks a little like a house with the oxygen at the top of the roof. And so we start numbering our carbons over here. Carbon number 1 has the nitrogenous base attached to it. Carbon number 2 must be important because I've given it a green, a different color. Carbon number 2, by the way, has a simple hydrogen atom attached here. Carbon number 3 has a hydroxyl group. Hydroxyl groups are more reactive than just a simple hydrogen. So this is important. This is our 3 end of our molecule. During elongation, this is where our new nucleotide will be added. Here's carbon number 4. And on carbon number 4, we have the chimney carbon of our little house-shaped deoxyribose. And on that 5th carbon, we have the phosphate group attached. So let's back out here and allow me to show you how we can do this conversion on our dynamic DNA nucleotide. So you can see here I only have a nucleotide, and we've got our nitrogen containing base, we've got our sugar here, and we've got our phosphate group. So let's focus on the sugar like we did in the diagram. We've got our oxygen at the top of the house. Here's our first carbon. And on our first carbon, we have the base. Here's our second carbon, which has a hydrogen atom attached. Here's our third carbon with our OH and our invisible hydrogen. Here's our 4th carbon. Here's our 5th carbon. And here's our phosphate group. The first thing you might notice is that I actually have to spin this to show you because it's not flat. So already we have a better model for understanding how this nucleotide acts in space. Okay, so let's focus on the sugar again. First carbon, second carbon. The difference between our deoxyribose and ribose lies in a simple change here. We just add a hydroxyl group, which you remember means that this is going to be a less stable molecule. All right, that's the simple change. So now this is ribose, making this a ribonucleic acid. And it makes sense that it's going to be less stable because remember that DNA is our permanent record and commands for how our genetic code produces proteins, but also RNA is a less stable molecule, and that's important because RNA has to be degradable in order to function in the way it should. We can do this with our other nucleotides. So we've got our cytosine. Here's the deoxyribose sugar, carbon number 1 attached to the base. Carbon number 2, I'm going to add the OH group. There we go. And there's our phosphate. So now this is the RNA nucleotide for cytosine. We've got adenine. Here's our sugar, carbon number 1 with the base, carbon number 2. Is now ribonucleic acid and the phosphate group. And then we have our final nucleotide. Here's thymine. Did you just say there's no thymine in RNA? So we're going to have to make a couple changes here. Let's see. So we've got our deoxyribose. Here's our first carbon with the thymine base on it. And then we have our second carbon. And again, we're going to add the hydroxyl group there. So first carbon, second carbon. Boop. There it is. Here's our hydroxyl group. Now that we have our hydroxyl group on there, this is a thymine nucleotide with a ribose sugar. How do we convert this to uracil? We just pull off this little thing group. So this methyl group, when it comes off, a C with its associated hydrogens, when that comes off, we now have a uracil. All right, so now we have our 4 bases. We have uracil, we have guanine, we have adenine, and we have cytosine, all as ribonucleotides, and now we can do transcription with our kids. The final thing I wanted to show you is ATP. So I'm going to share my screen again, and as I do so, you will see that we have ATP here. And generally, we take our students' attention to the 3 phosphate groups, but I'd like for us to focus for a minute again on the sugar. And what do you notice about that sugar? Is that deoxyribose or is that ribose? You're right, that's ribose. And it's important that it's ribose. These are less stable and more reactive functional groups than the simple hydrogen that you find on this second carbon for deoxyribose. And so if we wanted to use our nucleotide for adenine and convert it into ATP, that's fairly simple to do. I'm going to come back here and show you again our nucleotide. So we have our adenine base. We have our deoxyribose sugar, which I'm going to convert to that less stable ribose by just adding the OH group right there. And now we're going to focus on adding some additional phosphate groups. All right, so I'm going to add phosphate here. Now we have 2 phosphate groups. I can spin this around a little bit. I've got my ADP. Add 1 more phosphate group on here. Here's our last phosphate group. Now we have adenosine triphosphate. Again, not flat and moves around in space, all right, which is more accurate than our flat model. Gives our students a better idea of how this works. So what do I do then? I generally have my students build these in pairs, which is helpful, but then we really need to talk about why ATP is used as the energy currency or one of the energy currencies in our cells. What happens when something is phosphorylated? So at this point, I like to do a lot of turn and talk. I would have a turn and talk activity where the partners who built these, one student would take the molecule, pop off that last phosphate group, and phosphorylate their partner. All right, so Dan's over here. I'm going to phosphorylate Dan. And what happens when Dan takes this phosphate group? Well, Dan would need to come back and tell me that phosphate group might make him a little less stable. He's feeling less stable and more reactive. Or maybe it would cause a shape change that makes him more likely as a molecule to react. So that's one of the ways that I use the dynamic DNA nucleotides to demonstrate cell energetics.
Now that Karen has gone and made me less stable and I guess a little bit more energetic, what we're going to try to do is she showed you a lot of the ways that we use dynamic DNA to show the chemistry and to show a lot of high-level stuff. But I want to take it down a moment. And I want to talk about how I do this with my high school students and how we start out playing with the dynamic DNA. And one of the issues is playing with it. Get the students to play with the models and to see what's happening. Rachel's going to be putting up on your screen right now how to pin Karen's screen, so that way it fills up, so you're not looking at me, you want to see the models. That's what we're really here for. I know that I'm working on an iPad, and I found before that if I clicked on the image of the little image where you can see Karen's hand on the iPad, it switches between big, huge me back to what you really want to see, which is the models that are there. So my students would be, I would give each one of my students a particular nucleotide. And I'd say to them, what do you notice? What do you wonder? What do you see out there? And one of the things that they would notice is, as Karen said, it's not flat. It's fabulous. It comes out at you. It's got 3 dimensions. Then they might notice that there's the different colors on them. And probably the best thing I would ask them to do is start taking a look at their neighbors. What do you see that's different between yours and your neighbors? And one of the things that they'll start to point out is mine might have 2 rings and my neighbor has 1 ring, or they'd probably say something along the lines of circles because they're not in the areas of rings yet. Another thing they might notice is there's magnets in the middle, and some have 3 magnets in the middle and others have, for some reason, 2 magnets in the middle. And we saw in the last that these magnets are going to act as bonds and such, but we just want our kids to explore, play a little bit with this. And then you might notice that it's somewhat flat in the middle too. And it's kind of perpendicular on the sides. And so what might be going on with something? And of course, the kids are gonna play. As you saw, Karen started playing and seeing, do these magnets fit together? I said, sure, go for it. See if you can find how these magnets fit. Woohoo. Looks like something fits, or maybe not. Try to go around the room and see what fits. There you go. That's one that fits perfectly. And so the students start realizing that these things seem to come in sort of defined pairs. And they don't know anything about Watson-Crick base pairing at this point. They're just playing with magnets. And you might even ask them, do you notice anything about the pairs? How there's always 2 rings on one side and 1 ring on the other, or 2 circles on one side or 1 circle on the other. After that, you might say, well, great, you got these pairs. Can you do anything else with these base pairs? Can you see if we pair them up other ways within the classroom? And so they'll start playing and see what other combinations that can be made. Some will work, some will not. And then we see that they start forming the base pairs into some sort of ladder combination. And then they realize that, wait a minute, they could even start twisting a little bit and bending and changing shape. And some students are going to get a little rambunctious in the classroom and start adding more base pairs to each other. And normally I'd say to them, you know, hold off a little bit. I don't want you sharing this information because we want the students to discover. But as you start adding more base pairs together, Karen's going to try to scrunch it down a little bit, and you'll start seeing a structure that we've seen before, the double helix. And the students get so excited when they start seeing this. It's like, gotta let everybody discover this on their own. And I would say again, what do you notice? What do you wonder? And they would start to notice that, well, all the reds and yellows are on the outside, the whites and blues are in the middle. They would start to notice that there's this wide area on one side and kind of a scrunched in area on another side. And so this isn't, we haven't really talked about the major groove and the minor groove. We didn't get into any of the chemistry that Karen mentioned before. We got them excited because they recognize a double helix. We got them excited because they're playing with the models and saying, wait a minute, they fit together and there's some sort of rhyme and reason, and we could see what kind of questions are going to start coming out of this. So in the next little quick video that we have for you is we're going to be looking for, now that the students have this and start playing with this, what kind of structures, or I'm sorry, what kind of questions can come out. So I would start this play with my intro bio students. And then when they share up the screen and you see the movie, you'll see that these are the things that my advanced biology students would come out. And I want to emphasize again, this is coming from my students. This is not coming from, you know, where do I want to take it? What this is coming from is the students making connections.
Karen and I said it right at the beginning that what makes dynamic DNA such a great model is some of the questions that it promotes. And so looking up on the screen right now, you see how the dynamic DNA is twisting and turning, and having the students play with this brings up questions. Can it compress around the nucleosome? How does it compress around the nucleosome? Do you have some nucleotides are getting closer, some are going to be more exposed? Is the sense strand going to be something that's more on the outside of the nucleosome or the inside of the nucleosome? Is there going to be any steric hindrance that's going on within there? Can we elongate the molecule? Is that how expression occurs in some cases, where an enzyme will come in and pull it apart or maybe compress it down? How do these forces work within the actual DNA molecule itself? Well, we can model it and we can see how these things are going. The second thing that we would look at and see is RNA. We can build the RNA, and Karen showed us how we can put all these things together. Well, I made a little bit of a chain of RNA here. And in this chain of RNA, take a look in this spot here. You can see all these oxygens in a row. That's going to make it really unstable. That's going to be a problem. It's one of the reasons why RNA is not around for as much. And so then it brings up the question, well, then how do we come up with all these major structures that are made out of RNA and the three-dimensional things that are going on within there? So there's chemistry questions that can come up. And then we get to the really interesting stuff. Now, what I'm about to show you is stuff that I was modeling out for my students, and then my students started asking these questions. So these aren't my questions, these are my students' questions. So we see that we have a replication fork, and I put an RNA primer on there because we're always talking about an RNA primer, and I could model it with dynamic DNA. And we have the free 3' hydroxyl. And you can see the cartoon image of the free 3' hydroxyl up that we show in so many of our models. Well, why is it important? What's going on with that? Because that's where the new nucleotide can attach. So the replication fork pulls apart, and we could see DNA polymerase coming in. Now, this is a little bit corny in the way that I have it modeled, is the DNA polymerase would be clamped on and would be working with this, not something that's marching in from the left, but it helps the students to understand. And so we see the DNA polymerase marching in, and you can see all the phosphates now are getting really close to that free 3' hydroxyl. So that hydrogen on the free 3' hydroxyl is going to be important, and all those oxygens in the phosphates are going to be important. And we can see exactly what's happening at the active site right now of DNA polymerase. And as the active site pulls it in closely, you're starting to have the adenine matching up with the thymine, and the phosphates are getting really close to the free 3' hydroxyl. And we can see it here, is they're kind of overlapping with each other. So what's happening? Well, you could also see that in the way my model is it's causing the nucleotides to kind of bend and move a little bit. Well, is that real? What's happening? What's the chemistry that's coming on? These are the questions that my students were asking. And so it brings out a little bit of wonder. It's okay if we don't have all the answers on this. We want our students to say, hey, whoa, wait a minute. Is that real? Is that what we're looking at here? And so I pulled it apart a little bit so you can see the interaction. And of course, at this point, we pull off, and then my students were like, well, what do you pull off? Do you pull off a monophosphate? Do you pull off a diphosphate? And I was like, oh, I don't know. In my whole teaching career, I had always said, well, it's a dinucleotide triphosphate. It comes in, it creates the covalent bond in the backbone, which we can see this covalent bond forming here. And we can see that I pulled off a hydrogen and I've got a diphosphate. I had no idea that that actually happened. My students were asking me that question, saying, well, what does happen in that case? And I was searching for answers. So I had to go back and do a lot of literature reading. And that's okay that I didn't know that answer, that my students came up with that question. My students were asking the question because we had done ATP and we had done glycolysis, and everything was about, well, if I phosphorylate this, where's the hydrogen going to go? Where's an oxygen going to go? Where are all these things going to go? And my students were looking at the same way, cross-cutting concepts. This isn't just DNA. This is biology. And everything works together. And then, of course, it turns out it is a diphosphate that pulls off. Whoa, who knew? I sure as heck didn't. And apparently the hydrogen, it becomes an acidic situation. And so the hydrogen is free within the solution. So where does the phosphates go? What happens with this? And you'll see here that I brought in a new enzyme that I learned about called pyrophosphatase. And pyrophosphatase is going to come and break apart those two phosphates. And this is a highly negative Gibbs free energy, which is going to help drive the reaction. So you don't have to go into all this. I would never do this with my intro bio students. I would want to see my AP Bio students start questioning about this. And now that I could bring up the huge negative Gibbs free energy change, we can start talking about this in the same language that we talked about within cell respiration. Cross-cutting concepts. How is this all working together? What are we working with? This is what makes this such a great model, because it leads to more questions and more questions and more questions, and that's what we want to see.
And so as we've been very specific so far, zooming in on the dynamic DNA, we mentioned at the beginning that we also wanted to look at the flow of genetic information kit, which is kind of a zoom out. So we could use the dynamic DNA to demonstrate replication and transcription if you have a classroom set. But it's important that if you zoom out, you can start looking at the role of enzymes. You noticed on the screen, I had to write in all the enzymes, but with the flow of genetic information, you're gonna see that we have these enzymes that are part of the model. So Karen's going to take us through an introduction to bring us between the two models.
All right. So one thing that I often forget to do with my students is kind of bridge the gap. I'm sometimes make assumptions that they understand when I move from one model to another, that they're moving with me, but they don't have the framework always to move between models. So this activity, which is available on the 3D Molecular Designs website, was designed to go along with the Flow of Genetic Information kit, the Dynamic DNA kit, and the Discovery DNA kit, which Kim Parfit will talk about in a later session. You can see here that we have our flat nucleotide, our dynamic DNA nucleotide. We've got two new nucleotides. And one of the things that is important is for me to help bridge that understanding. So this is kind of a really neat little interactive that you can use as a hybrid activity, or you can allow your students to use it as a practice activity, or you could just use it in your direct instruction if you need to. So I've labeled the nitrogenous base on our textbook model. You can also go to our dynamic DNA model and show the base here. And this is going to be our new nucleotide that we're going to use to zoom out, and I'll talk about that in a minute. And I'm not going to worry about this one right now. So you can see every atom here, we can see the spacefill model here, and when we get to this flow of genetic information nucleotide, gosh, it looks super simple. And so we'll have to talk about its utility here in a minute. Here's our backbone, our sugar phosphate backbone on our textbook model, and our sugar phosphate backbone on our dynamic DNA model, and our sugar phosphate backbone down here on our foam pieces for the flow of genetic information. So when we look at these three models, what you're seeing here in our new kind of zoomed out model of a nucleotide are some different emphases. So what we've got here at the top of the foam model is a specific cutout that will allow, we'll see this in the next slide, that the complementary bases will be allowed to bond together, but for instance, a C and a T would not. So showing every atom here would distract from our purposes, because we're moving from structure to function here. So we've got the base pairing, because that's going to be important. We also have kind of an arrow, and obviously our nucleotides are not equipped with arrows. That would make things super easy for our scientists. This actually is rather a meaningful model instead of a structural model. So let's bridge that gap here. So we can take our 3' and kind of label our three nucleotide here and our 5 carbon here. So this is the 5' of our nucleotide, even though the 5 carbon is here at the top of the chimney. And we go to our dynamic DNA. Here's our 3 carbon here with the oxygen attached. We've got our 5 carbon here with our phosphate attached. And then we have, going down to our new model, our 3' and our 5' here. And so now you see the reason for the arrow. The 3' is where we're going to add the new nucleotides when we have chain elongation during DNA replication and during transcription. And here's our 5'. So this is where the prior molecule is going to be plugged in. So now we have a functional model rather than a structural model. And we can show the base pairs. So the actual activity has more slides than this. I just chose these 2 because they suited my purpose in this presentation. So in this, you can see that we can take our base pairs, and actually I should start with our dynamic DNA base pairs. So our guanine is going to kind of pair up with our cytosine, and you can pretty much snap them right where they need to go here. Pretty close. And here we have our adenine, which needs to have a kind of a flipperdoodle. So flip that guy around, and we can snap that into place right there. And so these are the ones that we've already played with. So now we're going to transition to our guanine and cytosine foam models. So I'm going to put this guy right there, kind of line up with the dynamic DNA nucleotides that we've already placed. And I'm going to flip this guy around. Whoops, wrong way. Flip it around this way. And of course, you know, your students will probably do this a lot better than I am going to. There we go. All right. Getting there. Aha. So then again, and I've actually pulled the labels for my own purposes from the prior slide. And again, I can label my 3' here and my 5' here. 5' whoops, goes down here. You can add a 5' here so that we can highlight the anti-parallel nature of our DNA. Right here. All right, so there we go. We've got our bases, and we've kind of scaffolded the transition from our dynamic DNA model when we talked about structure. Now we're going to zoom out and talk about function. So that is the end of my screen share. I'm going to pop right back out of there. And we're going to move back into Dan, who's going to talk to you about how to use that flow of genetic information kit for DNA replication.
Before you press play, Chris, I just wanted to bring up, this will be a very short little video, and then we're going to try to have some audience participation. And we've been in this for 45 minutes, and the video is going to be very short. And during the audience participation, there's definitely a built-in bathroom break. I was supposed to say that right from the get-go. So just in case you're wondering at this point where we're going to go. So I think this video is about 3 minutes. Then we'll get into the audience participation, which will have the built-in bathroom break. So whenever you want to press play, please.
what we see here is on the left-hand side, the dynamic DNA, and on the right-hand side, we see the full models from the flow of genetic information. Remember, all models are wrong, some models are useful. Each one of these models is showing particular things with its particular strengths, but they can't show everything because which one is the real DNA, which we can't quite see. So the DNA on the right-hand side is coming into the helicase where it's going to be split open. On the left, you're going to see how the double helix starts to untwist. And so the double helix is completely untwisted at this point on the left, and the helicase is breaking it open, so we're exposing the base pairs, which we now will do on the dynamic DNA side, where we have a replication fork in both cases. Now, the way I handle this is I really like to show the RNA primer. And so on the dynamic DNA, I could build that in with actual RNA pieces that we've seen. And on the flow of genetic information, I use the RNA pieces from protein synthesis. I just have the students build on a couple extra primers, a couple extra nucleotides as primers to show how you have that free 3' hydroxyl that you need to be able to have replication to occur. Polymerases are now going to hop on. I can't show that with the dynamic DNA, but I can within the flow of genetic information. And you see that the active site shows that area where we need the free 3' hydroxyl. Nucleotides are going to come in. On the left-hand side with the dynamic DNA, I just have a free-floating nucleotide carried in by the labeled DNA polymerase. On the right, I still have that polymerase there, and I can start sliding the nucleotides in. So each model is showing different things that I can now have the students expand out or zoom in for particular ideas that they're looking for. And of course, the nucleotides are integrated, and we can expand out. Certainly, the flow of genetic information kit is going to show Okazaki fragments much better than the dynamic DNA kit.
And so at this point, we're going to try to have some audience participation because, you know, as teachers, we like to play, you want to try, and of course, we wish we were all there in person. We sent several people the student modeling kits, and so you should have received that already. And we also had instructions in there of how to make a particular set of DNA that we're going to now try to replicate. So what we'd like you to do is, if you haven't done so already, please construct your template DNA sequence, which they're circling on the screen right now, and it should look like that picture. If you have done it, and even if you haven't, you see that there's a 5-minute timer up there. We are going to completely mute and shut down for the 5-minute timer, so that way you can set up your DNA, and then you can, if you have to run to a bathroom or something like that. Also, during this break, raise your hands when you're ready to turn on your camera, because we'd love to see what you're modeled. We'd like to have some sort of like Hollywood Squares where we all hold up and show all the different things that we've done, and that also gives us a chance to see all of you. So again, 5 minutes, please go ahead and make the models up on the screen if you haven't done so already, and if not, enjoy your break.
So, what we have next for you is an opportunity to kind of see how the student modeling pack works for transcription. And after that, I'm going to take you through translation. And the translation, well, we'll talk about that more after this video is over. So here's transcription. Go ahead.
Right now, we're going to start talking about how we can use this nucleotide student modeling pack to demonstrate or to model transcription. So in the lower left-hand corner of my screen, you can see that I've set up the student modeling pack with the RNA polymerase and the pre-built strand of DNA that we made during our DNA replication. And it's going to enter from the right-hand side. You might notice that I've done some modification here. I've taken a little bit of the foam that I popped the DNA nucleotide adenine out of, and I superglued it over here to help facilitate the opening of the DNA nucleotides. And also I've done the same thing over here, and you'll see why in a minute. So I've got my DNA lined up here, and I've got my RNA polymerase here. Everything's all set for transcription. When we move to transcription, we need to make sure that you understand that we've differentiated between the DNA model pieces and the RNA model pieces. So these round pieces are the DNA, and you'll see they're circular. And we've got these RNA pieces that appear to be square, and also in addition, we've got a uracil instead of the yellow thymine. Another thing that you're going to notice about these is that your bonding sites, the hydrogen bonding sites, are unique, so that theoretically your students will be unable to bond them inappropriately. All right, so the guanine will bond with cytosine, the adenine will bond with thymine, as you noted before, because of the shape of the hydrogen bond model. Same thing goes with the RNA nucleotides. So our DNA to RNA nucleotide, adenine to thymine. You can see right here that the hydrogen bonding region is complementary. So as we move on to the next one, I've begun to advance my DNA over to the RNA polymerase, and you can see that I have sticking out here my 3' end, right, my active end, and down here the strand is proceeding with the 5' end on the front side. And I begin to move. That's where this little piece that I've kind of engineered here helps to split this apart. But you'll notice that we've got a thymine approaching what we've labeled here as the active site. So once we get to this active site, we can start to bring in our RNA nucleotides. Those RNA nucleotides will fit right in the active site. And because we have this active site on the template, it guides students to be able to make sure they remember the anti-parallel nature of these nucleotides. So they're going to place an RNA nucleotide that is complementary opposite of the base. And we're going to continue to advance this. And you'll see that as we add these nucleotides, these RNA nucleotides, this little piece that I put in here is going to start splitting apart that, that RNA transcript. It's going to be starting to move across this land bridge, and students really like that. I think it's kind of cool too, because this RNA is going to be only temporarily bonded to its template DNA and eventually be peeled off so that it can go to the ribosome. So here we are further along, and now you can see that we actually have the DNA coming back together after it's been funneled through this little piece of the RNA polymerase enzyme. So we have the RNA transcript peeling off, and we have the DNA rejoining, and it almost happens naturally. It's very nice. Eventually, what you're going to see is that you have your original DNA molecule, and it comes back together and it can be copied again. You can have students flip it back over and make another copy of the RNA. Here's the RNA transcript. One note is that I like to have a little bit of leader RNA, some untranslated RNA at the beginning before your AUG start codon, and I like to have a little bit after the UAG stop codon, or whatever stop codon you choose to use in your situation, because that allows for students to understand that when we get to translation, the ribosome is going to kind of scan the RNA and find that first codon so that you can do some frame setting, and it also sets them up for understanding frame shift mutations later.
Now, we had hoped at this point that we would also have students be able to build this out, because playing with that ribosome is really, really cool. Sorry, not the ribosome, the RNA polymerase is really, really cool, because what you would want is to see that bridge. And what's also kind of interesting is the students will start asking you, is this real? Is this just some sort of thing that you put in to make the model work? And from what I understand is, if you go into the protein data bank or whatever, you can see how this all works. We're not trying to just pull one over on the students. We're trying to get them to see the wonder that's actually in the real molecules themselves. Hopefully you're all going and clicking on that link, and you'll get these sometime next week, because I can't emphasize enough how much fun they are to play with.
So at this point, what you would do next would be translation. And the student take-home models don't have a mechanism for translation. So I'm going to switch to the OG, the original kit, as the kids say. Maybe they said it last year, I'm a little behind. But the translation portion of this, I'm going to show you how the original kit works. And then Dan's going to show you a solution that he has, his students modeling translation with the student take-home packs.
Now we're on to the part of the kit that is actually part of the original Flow of Genetic Information kit, which we affectionately call FGIK. And here you see a ribosome. This is not included in the student modeling kit. However, Dan has a really brilliant idea for how students can model this part, this translation piece of protein synthesis. So in the original, you have a mat that is about the size of 11 by 8.5 sheets of paper. And you can see here that, again, we have some foam pieces that are going to help guide the process of translation. And you have your E, P, and A sites. I call this the eject site. I call this the primary site, even though that's not actually what it stands for, the peptidyl site. And this is the amino acyl site. I call it the add site to help my students. Your RNA transcript is going to enter with its 5' advancing through the ribosome and its 3' in the following. Meanwhile, off in the cytoplasm, you have some transfer RNA molecules and you have amino acids. So these amino acids and these transfer RNAs are going to bond temporarily so that the transfer RNAs can carry their amino acid cargo to that ribosome, where they can settle in on the messenger RNA transcript where appropriate, as deemed by their anticodon. So here I have the first one. Here's our AUG codon. You can see I have this little untranslated region here. It's kind of like a leader. I tell my students it's like a lot of them are working in Target or in other places where they use a cash register, and they know they have to feed a little bit of that cash register tape into the machine. And I kind of tell them that this is a little bit like that. It helps the ribosome to hold on to that messenger RNA transcript. And so when it finds that first codon and it lines up with the P site, that first transfer RNA is assisted here, carrying the appropriate methionine amino acid. In comes our second transfer RNA, carrying its cargo. In this case, it's isoleucine, and we're going to watch right here as we get a bonding reaction between that methionine and the isoleucine. This is a covalent bond that tips the transfer RNA into less stable situation, and it shifts over to the eject site. Meanwhile, we have our third transfer RNA coming in. And so now this initial transfer RNA is going to be able to leave. We again are going to have additional transfer RNAs leaving, additional transfer RNAs coming in, forming these covalent bonds. And you can see that the peptide sequence is going to start moving out of the ribosome. And another thing at this point I can tell you is that I like to remind students that this would be like a cross-section of the ribosome. They're only really seeing the front of the ribosome sliced away so they can see what's happening inside the ribosome, because this ribosome is a three-dimensional structure. And so on we go until we get to this very last codon. It's actually not the last set of RNA nucleotides, but you can see the stop codon advancing into the A site. Transfer RNA, and that allows the release of the polypeptide chain. And so that will either go into the cytoplasm or the endoplasmic reticulum so that it can be folded appropriately due to its properties, and also it with the help of some enzymes.
And so as Karen had mentioned a few moments ago, this doesn't come with the student modeling kit that hopefully you'll be getting this week. And I kind of want to emphasize that Karen and I, she's a college professor, I'm a high school teacher. This is the stuff that we use in our classrooms all the time because we love it. It's versatile. It does so many cool things. We're not the people who work in the company. We're the users of this. And one of the reasons why I love it so much is because it's so easy to play with and adapt to other areas. I was frustrated this summer because, you know, my students, they're in remote learning and we've got all these issues and I can't use my toys. I can't use the flow of genetic information kit. So I thought, how can I play with this at home? And what you'll see in the upper right-hand corner of your screen right now is I just traced something out on a piece of paper. And I put in the E, P, and A sites, and of course I labeled it a ribosome. The student modeling kits that you get come with the nucleotides. So the students can do this, and then I can you know, show them how we can make a at-home model. The second picture down, I figured out, well, I need to have a tRNA somehow, and I thought, well, what if I just made it with a piece of paper? And I made it where you have a site for a codon and an anticodon. And you'll notice that I actually didn't write anything because my thought would be, let the students write it in. Let the students kind of discover what should be going on in this area. Not a worksheet, a model that they can go stepwise throughout all of this. And I also used Post-it notes where as amino acids, and I drew in the actual structure of the amino acid, at least the carbon backbone, so they can start seeing, again, relating from their textbook structures where they can see the amino side and the carboxyl side. And then, of course, you would have the R group, which shows that that's methionine. That's going to be up there. So the students can relate going from codon to the anticodon on the tRNA to a paper Post-it note that brings in the amino acid. And of course, this is my crazy idea. This was something that I thought through because I kind of have this vision of what's going on. What I love is when the students think they know something and they're learning it in class and they're going through the textbook, and then you say, okay, guys, you got the stuff, model it. What can you come up with? What can you play with and to show? And I'm seeing that, you know, somebody was saying you can make a stop motion version of this, and yes, you can. That's where it gets really fun, is you say to the students, go home and make a stop motion tonight. Play with this, see what you can do, and let's share it out. And you got to have it in kind of a safe zone, because what you're hoping is that the students you'll uncover areas where they have misconceptions. And wow, do they have misconceptions. I always thought, I showed everything correctly. I demonstrated it. And then the students start playing and you realize, oops, I didn't realize it came across that way. And that's what the models show. You get to see what the students didn't know they didn't know. And so if the students make an incorrect model, fine. Let's use it as a teaching opportunity to go. And they're going to be a heck of a lot more inventive than Post-it notes and little strips of paper to be able to show things that they have in their own houses and stuff. So I wanted to put this together so you can see my concept of it, but I think the students would do a lot better knowing the creativity of my students. And lastly, we're getting to the end of our prepared remarks. As you can see, all of the different standards that are up there, and of course this presentation will be out there, but it covers lots of different standards throughout. And you can see we have different ways that you can order this as well. Please be sure that you have the show special discount code CASE20 if you're going to go and try to purchase any of these kits. And of course, the student modeling kits are great because if your students are remote or if they're in hybrid, you could have one per. It's for the cost of an individual lab. I was able to go out and buy sets for all of my students. Okay, and Karen, if you wanted to say anything before we get to questions.
I just wanted to say thanks very much for coming to our presentation, and we're excited to hear whatever questions you have. Both Dan and I have been using these models in our classroom. I use them with my high school students, grades 9 through 12, and I use them with my college students too. So I'm excited to hear what you guys have to say or any questions that you have to ask.
Good job, Karen and Dan.
Thank you. If we still have people out there, I do want to say one thing, that the kits, I've been using this stuff for 11 years now. I've been a junkie of 3DMD, and the kits hold up, and they hold up to high school student mega abuse. So it's not something that you're investing in, oh great, I got one shot out of it. It's something that I get to grow with year after year after year.
Another thing is that, just real quick before we go to the questions in the chat, you can disinfect these models. There are instructions on the website for how to do so. Thank you, Stephen. We hope that you kind of dig into some of these models. I can't wait for you guys to get your modeling packs next week and start playing around with them. It's interesting to see what students do know and what they don't know and how it comes out in the modeling. Some ways that we can get our admins to get these for all the students. Heather,
I have a suggestion.
I knew you would.
So one thing that we do have, and that's with our partner, the MSOE Center for Biomolecular Modeling, they have a lending library. So you can get a suitcase of these models and actually get them in your school and, you know, get them in the hands of some of your students and have your admin drop by while your students are using these models. So there's that, and even the student modeling pack that, you know, we're happy to send you now, you'll be able to see that. And just to compare, I'm going to see if I can do something fancy and switch to my overhead camera. So this is the student modeling pack to be able to do replication, that what we're showing or sending you guys. This is the classroom kit. This is the original, where you can see that it's much larger. It has the real bridge, and that, you know, as you slide things through, oh, you can see, then as you're going through, things actually do slide and work, and you can just keep making those base pairs here in the active site.
So
okay.
One thing that my students have a hard time like doing that transcription thing without making choo-choo train sounds. But yeah. Another justification would be the modeling is a cross-cutting concept, or a practice, sorry. And also, you know, if you buy kits that are expendable each year, such as like a DNA extraction kit or whatever, you have to pay for that every year. These kits last year to year, so that might be another argument that you could use to get yourself some funding.
I could say from experience, having your admin walk in when you're using these kits and the kids are having so much fun and then they could intelligently speak to the admin about all the different parts of the model that they love and that they can demonstrate. And often the admin has no idea what the students are talking about because they don't necessarily know the science. And it's the modeling is wow, the student interaction is wow, and the student knowledge is wow. And then you got the admin hooked whenever you come to say, I need to get a couple of these for my classroom.
And Rachel did post another link about some other funding ideas that we've put together. And Diane was sharing that there are some academic papers that we've linked to on our website that talk about how modeling is really effective for your students. And in some of them, it's really helped bridge the gender achievement gap between boys and girls. So it really helps bring some of our young women who may have their brains may not have developed that spatial reasoning as much yet, and so the models really help to bring them to the same level as the boys. And then everybody actually increases, but the girls, young women have a larger increase.
I used a different model with my students yesterday. I told you I'm a junkie with all this stuff. I have lots of the kits. And my students stayed after class yesterday, A, telling me how much fun they had modeling, and B, they were so grateful with the hands-on that this wasn't a video, this wasn't a, just draw a picture or whatever. It was hands-on, touching things, moving things around, and they loved it so much and probably appreciated it more because of all the hybrid stuff, or I'm sorry, all the remote stuff that they've been forced to do. And getting the student modeling kits into their hands to do socially distant work has been fantastic.